Hi, everyone. Welcome to everyone joining us in Zoom and in person today. It's a real pleasure to have you here for one of our HBI 101 talks. Today, it's my pleasure to introduce Dr. David Silbersweig. Dr. Silbersweig is a neurologist and a psychiatrist who works at Brigham and Women's Hospital and Harvard Medical School. He is the chair emeritus of the Department of Psychiatry at Brigham and the Stanley Cobb Professor of Psychiatry at Harvard Medical School. He also co-directs the Brigham and Women's Hospital Institute for the Neurosciences, and I'm excited to say he's joined our HBI steering committee. Dr. Silbersweig did his undergraduate work at Dartmouth, where he studied philosophy. He then went on to medical school at Cornell. He trained at New York Presbyterian Hospital, Weill Cornell Medical Center. Then he studied functional brain imaging in London, came back, started his own functional neuroimaging lab at Cornell, together with Dr. Emily Stern. He was then later on recruited to Harvard to chair the psychiatry department, as I mentioned, at Brigham. He is one of the pioneers of functional neuroimaging work in psychiatry. His work involves developing new imaging techniques to understand the basis of various different mental health conditions, the neural circuits that are affected in those conditions. This has involved developing new methods in PET and MRI imaging. Just to give a few highlights, he has studied abnormalities in the modulation of the frontal limbic system, which is important for emotion regulation across a variety of different psychiatric disorders. Worked on finding final common neural pathways that underlie different clinical phenotypes in different psychiatric disorders. Excitingly, did some of the first studies finding the brain abnormalities that are associated with hallucinations in schizophrenia, with tics and Tourette syndrome. Has also had a lot of contributions studying neural circuits that underlie depression, post-traumatic stress disorder, borderline personality disorder. And overall, his work can be thought of as systems-level neuropathophysiology. The ultimate goal being to develop novel, targeted, biologically based diagnostics and therapeutics for a range of different mental health conditions. And beyond his research, he's really involved in a wide range of community building, educational efforts, really excited about connecting sciences to humanities and a variety of different disciplines. He puts it as transdisciplinary initiatives excite him. So that's even more why we're happy to have him on our steering committee. And before we get started, I just wanted to say a couple housekeeping things. We have a small group lunch for graduate students and postdocs after this. If you have signed up, please come to the front here. And please ask questions during the Q&A, but if you want to ask them individual questions, wait till we get to the lunch, because a lot of times people are so excited, they just gather here, we never make it to the lunch. And if you have not signed up, but you are interested in participating, we do have a couple extra spots, so just come to the front and inquire. If anyone has questions on Zoom, feel free to type them in the chat at any time. Thank you very much, Dr. Silbersweig.
Thank you, Farzad. Can you hear me with this mic? Is that all right? Thank you for that gracious introduction and for your leadership in the Harvard Brain Initiative. It's an honor and really exciting to be part of it and to be thinking together with neuroscientists throughout Harvard and all the different schools to be able to advance biomedicine, in my case, and try to do something to increase our understanding of what's going on in the brain when patients suffer with mental disorders, which affect every single family in every culture and regardless of anything else. The morbidity, the cost, the suffering associated with these conditions is enormous. And we'll sort of unpack, quote, neurologic disease and, quote, psychiatric disease. I'm putting that in quotes because at the end of the day, what we're very interested in doing is identifying final common pathways of disease expression and phenotypic expression in clinical syndromes across all of neurology and psychiatry that produce disorders of perception, cognition, emotion, and behavior and relationships. And hopefully the aim is to then do something about that and to increase our understanding to the point where we may be able to translate that. And that, of course, is our aim. And it's a particular pleasure to speak with you today. I love speaking with people in the sciences and at different stages of their trajectories. I'm happy to speak with folks, in the lunch afterwards. A disclosure, I won't be talking about it, but some of the work that we're doing has spun out of the Brigham and Harvard into a company that's trying to translate this into the clinical aspects. But I'm not going to be speaking about any of the company's work or the like today. I love to start with this slide, with this quote, in fact, by Hippocrates. And it's extraordinary that it was written when it was written. And we might surmise that maybe he was observing people before and after head trauma, that somehow getting hit on the head changed your behavior in a different way than getting hit on the elbow. Although you might be upset after being hit on the elbow, it doesn't necessarily affect mentation and behavior in the way that something up here does. And just take a minute to, well, I'll just read it. I mean, and men and women ought to know, and everybody ought to know, that from nothing else but thence, from the brain come joys, delights, laughter, and sports, and sorrows, griefs, despondency, and lamentations. And by this we acquire wisdom and knowledge, and seeing, hear, and know what are foul and what are fair. And by the same organ we become mad and delirious, and fears and terrors assail us. That's wild, right? I mean, that he would have said that at that point. And in fact, that's the context for what we're doing. And similarly, this is Disease Man, an illustration from around the year 1400, which I love because they were trying to do what we're trying to do, which is to localize core mental faculties in the brain. And in this case, you can see, if you're classic scholars, imagination, memory, common sense, etc. And arrayed about the head are the maladies that accrue when those core faculties get disrupted. And so they were trying to do brain mapping. We're trying to do brain mapping. We'd like to think that we're doing it in a more empirical way. What I'm speaking about today is systems level neuroscience, which, with all the benefits and limitations thereof. The benefits are that it's the only level in neuroscience in one sense where you can directly correlate brain states and mental states in health and disease in a way that covers the whole brain and allows you to look at large-scale distributed networks and to push and pull them and to ascertain what's going on there. Now, of course, the limitation is that that's not the cellular and molecular level, but we have our friends in basic science who do the cellular and molecular work, and we work together exceedingly closely with them in this translational bidirectional relationship where we inform them, they inform us, and I'll give examples of that as well. And then on the other end, with folks in cognitive and affective psychology and neuroscience who study the phenomenology in that regard. And so this is very interdisciplinary work requiring interdisciplinary teams. And our lab, for instance, has everything from physicists and engineers, mathematicians, statisticians, neuroscientists, psychologists, psychiatrists, neurologists, and that's the fun of it. But it's also the challenge, but that's where science is these days, and it's big data with advanced analytics, as you'll see. So fMRI, which is the technique that we're focusing on today, really developed in the 1990s and is ideally suited to look at the interface between neurology, between mind and brain, to sort of literally and figuratively illuminate this circuitry that underlies these core functions and to identify the pathology that is involved when things go awry. I won't get into the details. This is not a methodologic talk, but suffice it to say, in an oversimplified way, that what we're doing is we're hitching a ride methodologically with our methods and technologies on the blood flow and oxygenation and blood volume changes that accompany increased neuronal activity in a localized area in the brain when it's active doing its thing within the context of about 1 millimeter and on the order of seconds. We use electrophysiology or magnetoencephalography to get to millisecond level, but they can't give you the same picture of the whole brain, if you will. And the point of this slide, again, without going into details, is that you really need to get into the biophysics and physiology of it, but the math and statistics is key in a very computationally intensive way, because what we want to do is pull out these tiny signals and to look for statistically significant changes that are associated with the mental states or brain states that we're interested in based upon our experimental design with control conditions or control populations, like in any form of science. And instead of like neuroradiology, where clinical neuroradiologists will look at a scan qualitatively with their eyes of structure of the brain, we're looking at statistical images of the function of the brain. And we're getting quantitative or semi-quantitative results to be able to test hypotheses or even do exploratory and data-driven sorts of analyses these days, which also are very exciting. I spoke about the translational chain. This has the arrows going in one direction. You can imagine them going in the other direction. When I was in New York, we had the great opportunity. I was co-PI with Bruce McEwen and Joe Ledoux and John Morrison, who are very major neuroscientists who've done seminal work in the neurobiology in rodents, fear and stress. And they would do in the center grant, they did the animal work. Liz Phelps, who's an incredible cognitive neuroscientist, did the work in healthy humans, and we did the work in patients with psychiatric disorders, although in this case, the Eisenberg study, we also studied healthy humans. And the bottom line is you can go all the way from, for instance, fear conditioning experiments in rodents and fear, freezing in rodents or fight or flight reactions, as we'll talk about in a second, are relatively preserved across different species, including us, although there's a lot else that has happened as well. And by studying, therefore, in the rodent, you can really get into the cellular and molecular. And Joe and Bruce have done extraordinary work there. And we're able to then be informed in our hypothesis testing by that, and in turn help them know where to look and to think about the models in the rodent work that would be applicable to the human disorders. And so we go all the way from being able to study the fear in rodents to threat responses in humans with activation of the amygdala here, areas in the middle of the temporal lobe, slicing the brain like this, and then to studying PTSD patients, post-traumatic stress disorder patients, where we wanted to test the hypothesis that this evolutionarily conserved system that is adaptive under some circumstances, if a lion is running after you or something, when in the face, if a person is vulnerable biologically and psychologically in ways that we can understand increasingly, the system gets perturbed to the point where it starts to activate on its own. And so the person experiences the physical and psychological aspects of fear and stress in a way that is out of control and that leads to a tremendous amount of suffering. I put these two slides together, or these two panels on the left and on the right. And if this were like a small class and we had more time, I'd say, what do you think the story is that I'm trying to tell by putting these two series of pictures on one slide? And to cut to the chase, on the left, it shows the relative conservation phylogenetically of the limbic and paralimbic cortices, whereas on the right, you see that the neocortex, and especially the prefrontal cortex, and especially in humans and Homo sapiens, is massively increased, right? And that's why, again, we can study some of these deeper regions and older functions in animal models, but they're still not adequate fully for understanding human psychiatric disorders. And for instance, in major depression, we need to understand everything from the fact that there's decreased sleep and appetite and sex drive, which are at the level of the hypothalamus and these deep limbic subcortical structures, these elemental homeostatic functions, appetitive and otherwise. On the one hand, that could be an aspect of major depression. On the other hand, you can have an existential crisis and suicidality and so forth, where, to have an existential crisis, arguably, you need neocortex to even have that level of representations about, even lack of meaning, let alone meaning. And this is why we need to understand the whole neuraxis and how it's wired up and connected in these disorders. Speaking about the neuraxis, I mean, and this is a famous diagram by Marcel Mezelam, and based upon Brodmann's work, where he looked at the cytoarchitecture of the brain and ascertained all these different regions that, based upon their cellular type and structure and connections and how many layers and distribution, could identify all these different areas that have come to be known as Brodmann's areas. And it turns out that the cytoarchitecture actually determines the transform and the computation that occurs in that neural tissue. And as we'll talk about in a second, it's all connected up. On the lower right, you see work by Jeremy Schmahmann and others in using diffusion tensor imaging, which is a way of imaging white matter tracks and axons in the brain. And between these local processing modules and then these white matter connections that connect them locally and at distance, we have the foundation for structural and functional connectivity. And these days, we're very interested in so-called connectomics in the brain, using advanced analytic techniques and imaging techniques to be able to then look and see how these nodes in these networks are functioning or failing to function, or communicating or failing to communicate in different ways that may be associated with important phenomena, whether psychologically or medically. And we want to understand how these systems are connected. On the left, you see just examples of some of these parallel systems that go from the cortex to the striatum to the pallidum to the thalamus and cortex. I'm covering a lot today, by the way, and I'm not expecting that you're all neuroscience students or that you've been exposed to this before. On the other hand, I want to really respect your intellect and give you a cutting edge view of what's going on in the field. So this is like a big download of a lot of information, and I'm trying to thread a needle at the one hand to give an overview, but to also give you robust details to give you a sense of the thinking and the progress in the field. So don't worry about having to remember details. It's, I could give a different kind of talk, but I found that this kind of talk by the end of the hour, while your head may be, full of all these things, or they may be out of your head right after, you at least will have heard them all put together like this, with enough rigor on the one hand, but enough overview on the other hand, hopefully, if I've done my job correctly, which I may not, and feedback is welcome. But on the left then are some of these parallel circuits that go through the brain, and each circuit subsumes a different function based upon a certain area of the cortex going to a certain area of the subcortical nuclei going back to the cortex, etc. And it's this feed forward and then in other modules feed back that's going to be really important in terms of how the behavior is modulated. And you can even think about this in zooming way out in terms of something where my mind is these days in terms of complex systems theory, and something called the free energy principle, where you can look at the brain as a complex system that essentially is a predictive organ that predicts its environment and acts in anticipation of those predictions and updates itself, if you will, in a way that is evolutionarily constrained. So having said that, what are some of the core systems that we want to interrogate potentially to try to understand psychiatric disorders because of the nature of the sort of functions that mental functions and behavioral functions that get disrupted in psychiatric disorders? So again, I'm sort of bucketing them here for heuristic purposes into sort of 4 domains that we'll quickly go through, and then we'll dive into some examples from our work and other people's work about how we're then putting the imaging methods together with the functional and behavioral neuroanatomy that we're learning from these methods, and then trying to understand the pathology in these diseases. Does that make sense? Okay, so let's start with and I alluded to it before in the work that we did with Joe Ledoux and Bruce McEwen, these threat responses. And again, to make a long story short, and at the risk of oversimplification, an area that has been really highlighted as being instrumental in a lot of senses of that word is the amygdala, which you may have heard about. Gets its name from almond. It's sort of the size and shape of an almond, and it's deep in the temporal lobes of the brain, in the middle front of the temporal lobes of the brain. And suffice it to say that it's been shown by Joe and Mike Davis and others that this area is absolutely critical for essentially pairing, even in one trial memory, a threat with a fear memory, which then allows the organism hopefully to avoid that in the future. So it's very, very important. It turns out that another area near there, the bottom of the hippocampus, which is a very important area for memory near there, is involved in contextual aspects of emotional memory. That's why you can remember where you were and what you were doing when something tragic or big happened, because your brain puts a time and place stamp with episodic memory on something that's emotionally charged, because that's adaptive for you to do. Although, again, it can get out of control, as we'll see. And then the medial prefrontal cortex is an area that's a higher order that sort of feeds down on those systems. There's also those systems project to there, but feeds down and helps to inhibit and modulate these systems. And all of these systems, we're talking today about functional neuroanatomy, but the neurochemistry and the neurotransmitters and peptides and everything else that regulate these systems is another key area of study. Bruce McEwen's work on stress and other people's has shown that stress is literally toxic to the brain. And cortisol, which is key, the cortisol system, the stress hormone, is involved in terms of its secretion from these fear systems and threat systems, going down to the hypothalamus and the pituitary, then secreting, then to the adrenocortical cortex, adrenal cortex, and cortisol is secreted. But importantly, Bruce found that there are cortisol receptors on the hippocampus and other regions, and it feeds back onto the brain. And stress can literally be toxic to the brain. And you see a neuron in red up here, a neuron on the left, which is healthy in the dentate gyrus of the hippocampus, where there's birth of new cells and neuroplasticity and neurogenesis. And you see on the right in a stress model in a rodent how literally the dendritic arterization is all shrunken. And so when they say stress is bad for you, you know, at least unmitigated stress, that's true. And increasingly, we're realizing too that stress and trauma and abuse neurodevelopmentally in kids and can really affect and set the set points of this system for their whole life in a way that may predispose to mental illness and physical illness, even when they grow up decades later. And with epigenetics, we're discovering now that even it can be passed to the next generation to a certain extent. So a lot of sort of implications of all of this, including really for public health and policy. This is just a slide to show that, you know, mental pain and physical pain have a lot of overlap in the brain. It's not just metaphorical. When you say I'm in pain, when a person, you know, is suffering mentally, literally areas in the dorsal anterior cingulate and anterior insula, and we won't get into the details, are convergent in mental and physical pain. Whereas other areas, like the ones in yellow in some of the sensory areas of the brain, are more specific and coactivated when the pain is physical and localized to a certain part of the body, of course. But these, again, are evolutionarily adaptive ways in which we as an organism, either psychologically and or physically, sort of withdraw and ideally, you know, stay safe when things are bad, to oversimplify, but again, that can get out of control. Whenever we're considering these systems, we want to think all the way from what we call the afferent or input to the efferent or output, all the way from how we sense things in the world to how we then, in the context of our internal states, appraise or interpret those things that we see, how we evaluate them, how we value them and weight them, then how we're motivated or not to act based upon that, how we initiate that action, how we monitor or change that action based upon changing circumstances, And this is all part of what we're interested in, this whole chain of feed forward and feedback in the brain. We talked about the negative threat withdrawal responses, right? Conversely, there's positive reward, positive emotion approach systems of the brain. Makes sense, right? And here, this whole medial forebrain, all the way from the deep areas that are evolutionarily older to these more neocortical areas and everything in between, are involved in generating action, motivation and action. And an area right in the middle there in the basal ganglia called the ventral striatum or nucleus accumbens, which is very rich in dopamine projections from the midbrain, but different projections than we see in Parkinson's disease that control motor circuits, is very involved with reward. And this area actually gets hijacked in addictions, which is why they are so difficult to treat. And they are true medical illnesses, that these substances of abuse literally hijack the reward system of the brain such that a person values the consumption or needs the consumption or to avoid the withdrawal symptoms also more than other things that would normally be and adaptively be rewarding, like family, work, school, etc., and things get disastrous. And this is a balance. It's a yin-yang, really, of approach avoidance. And we have to have a way of regulating this balance. And this gets to these higher order control regions. A sort of in-between region is the insula, which is deep in the brain, sort of enfolded between some of the lobes of the brain, that is, among other things, important for integrating a lot of this information from our body and social and emotional information as well. And the cerebellum in the back of the brain is not just for motor function like it used to be thought, but also, and our colleague Jeremy Schmahmann has shown, is also involved in cognitive and emotional function as well. And so how do we control all these things? That's where the prefrontal cortex comes in. And this massive amount that increased in our species, as we talked about before, and it's involved with a number of things that we call executive functions of the brain: monitoring, planning, addressing conflicting possibilities, evaluating higher order abstraction, etc. And there's different subregions within that. And those subregions include, on the bottom of the brain, the orbitofrontal cortex, which we'll see is important for social emotional control, among other things. The anterior cingulate and the medial frontal cortex, which is involved, as we talked about, in generating activity and modulating behavior. The dorsolateral prefrontal cortex, which is involved in some of these executive functions, like keeping something in your head and thinking through it and being able to monitor things. The ventrolateral prefrontal cortex involved in switching and inhibiting thought and behavior. And then the rostral prefrontal cortex, most in the front, the highest evolved, most recently evolved areas of the brain that are involved with higher order abstraction and insight, which, as you might expect, are really key for some of the things that we need to do. And then there are these systems that allow us to have a representation of ourselves in our body and as a person over time. And so the sort of spatiotemporal integration of representation of your own self, and then the representation of other people as well, something called theory of mind, and these areas in the dorsomedial prefrontal cortex and some of these higher order temporal parietal regions are associated with the ability to put yourself in somebody else's shoes, to even have a representation in your mind brain that somebody else has, or some other person has, their own intentions and emotions, etc. And these functions are sorely lacking in autism, for instance, where the awkward social behavior is not just, it's not obviously not the person's fault, but their brain literally can't represent that another person has mental states, let alone react to them or interpret them in real time and express and communicate appropriately socially. Okay, so all of these regions then come together to form networks that are operating together in space-time through oscillations in the brain, at different frequencies that connect activity through these systems that the white matter tracks and functional systems that we talked about before, even in the resting state of the brain. And this was a big discovery by Marcus Raichle and Mike Fox, who's at the Brigham, and who Andy Pine works with in the back there. And these systems in the brain are important even when the brain is just idling. It was almost a discovery that was right under everyone's nose that everyone missed, which is like, what's the brain doing when it's doing nothing? Well, it's never doing nothing, right? We're daydreaming, we're ready to receive sensory information, etc., mind wandering. And these are systems that are important for that. And then when we engage in a certain activity, they sort of turn off and go down in their activity, and other systems come online, like some of these executive systems in the frontal parietal top of the brain, in the executive control networks or attentional networks. And then these emotional networks that we've been talking about are sort of in between, and according to some theories, even mediate the switching or the activity between these networks. So it's very big now to be looking at the, quote, resting state with fMRI and these whole brain networks and doing advanced analytics to pull out the signals in space-time to understand what these patterns may mean. And what they mean is only as good, you know, if you're studying the biology, it's only as good as how fine-tooth a comb we use to study the phenomenology, the clinical features of what the symptoms are in the patient psychologically and behaviorally. So I'm not going to go into this, but the bottom line is the more precise we are in the exact behavior or mental state, then the more precise we can understand what the biology of that is. And unless we're fine-grained and nuanced in advancing the science of phenomics, you know, we're not going to be able to make the links that we need to make. And clinically, this becomes very important in what we call differential diagnosis and being able to figure out what's going on in the brain and then where in the neuraxis things may have gone awry. Okay, so take a breath, okay? We just did a sort of race through a lot of imaging and behavioral functional neuroanatomy. And now we're going to switch then. How do we use these tools and this understanding to then try to connect the biology to the psychology in health and disease? And how do we think about designing experiments and conducting experiments to be able to learn something that hopefully may be helpful for these conditions? So on the left in the upper part, you see, can you see my, I can't even see my, you can see the cursor? I can't even see it here. But on the upper left are some of these systems that some of these neural networks and functional systems that we talked about. On the lower right is a sort of cluster Venn diagram of how we might think of a current descriptive understanding of psychiatric disorders, which is to say imperfect. It's not biologically based. And there's a lot of heterogeneity within psychiatric diagnoses, which are defined according to something called the Diagnostic and Statistical Manual, DSM, and which is great in terms of defining syndromes and certain things, but it's not precise. And there's a lot of overlap too: depression, anxiety, psychosis, where, you know, mood, and so on. So what we literally, like, one way of thinking about, at least my career, and, you know, was the sort of science I've been part of, is trying to connect those two to understand that the biology underlying that, and to create a new diagram or a new taxonomy, sort of nothing less than a sort of biological, neurobiological foundation for a different understanding of how we can classify and understand mechanistically these disorders, but not eschewing the psychology or the psychosocial elements, which are absolute, and sociocultural elements, which are absolutely key, but understanding how the brain mediates those and those affect the brain and vice versa. And so, so we design experiments, and others do in the field, to be able to interrogate these systems and to test hypotheses or do exploratory analyses. Okay, so now let's jump into some examples. Take another sip of coffee. Take a breath. Okay. As I alluded to before, and again, I'm just cutting to the chase here, and I'm not going into the details of any of these experiments, but I'm happy to go into them offline. Long story short, and I know you're not necessarily all used to looking at brains, let alone which slice or orientation. Don't sweat it, right? But trust me. And Dr. Pine in the back, you want to suggest what these yellow regions are here?
In the amygdala.
In the amygdala, right? And these are these fear regions of the brain. And guess what? In a study that we did, where we gave trauma-associated words delivered in the scanner that people had to look at, and then neutral words that were matched linguistically and visually, but that didn't have that affective charge, and frankly, even other emotional words that were not trauma-related, we see, and then compare the brain and ask the computer, show us those areas that are statistically significantly different when looking at trauma words versus neutral words. We see the amygdala, and furthermore, we did a correlational analysis on top of that categorical analysis. And we then said, furthermore, if we have a group of PTSD patients, some of whom score higher on psychological distress, some lower, some score higher on physiologic distress, tremulousness, sweating and everything else, some score lower. And we use that as a covariate of interest against this difference of the threat versus neutral to give you a sense of how these experiments are designed. Lo and behold, these regions pop out like a Christmas tree of activation in the amygdala. Now, I'm not going to emphasize this part too much, but each time I show you a psychiatric disorder that has a symptom, I'm going to show you a neurologic disorder that has a very similar symptom to make a point, okay? And here you see temporal lobe epilepsy, and in a patient who had fear auras and who had experienced involuntary fear, didn't have, quote, PTSD or an anxiety disorder, had a, quote, neurologic disorder that affected the same region, namely the amygdala and ventral hippocampus, and guess what? They had the same sort of anxiety. So the key point, and I'm going to hammer this home and triangulate, and this was some of our work, is that it does matter for sure whether you have a tumor or epilepsy or genetic or neurochemical dysfunction, et cetera. But I'm starting now to talk about final common pathways of symptom expression, that if this amygdala-centric system, fear system, gets disrupted, whether it's by PTSD and neurochemical and experiential means, or whether it's by epilepsy or a tumor, you're going to have a fear-related symptom because that's the system that mediates fear, and you're disrupting that system in a reproducible way. Similarly, this is work that we did on hallucinations and schizophrenia. I won't get into all of this, let alone the experimental design, et cetera. This was actually a PET study, but and we superimposed it, a thresholded statistical image in color on top of an anatomical image of the patient's brain. And you can see this was a patient who was having visual hallucinations, and you can see visual association areas here in the occipital visual cortex, but his eyes were closed. He wasn't viewing anything, but the visual cortex was active. So of course he was seeing things. He's not crazy, quote unquote. He's just telling us what his brain is telling him, right? And then his auditory linguistic cortex in the left temporal parietal cortex was active, and he was hearing voices because auditory and language cortex was activated. Bunch of other things here in terms of why patients with schizophrenia may end up believing it and having emotional charge. I'm not going to get into that. Classic experiments in the 50s and 60s by Penfield, a neurosurgeon in the Montreal Neurologic Institute, in patients with epilepsy who didn't respond to any medications and needed to come to surgery and be awake, and they put in the electrodes to see where they could cut, et cetera. And when he stimulated the electrodes along those same regions of the brain, and then recorded for each electrode when they turned on the juice, what the patient said they were experiencing: voice, some cases music, depending upon which electrode, voice, familiar voice, right? So again, we're making the point, In schizophrenia, it's sort of auto-activated for pathophysiologic reasons that we're trying to understand. In this case, Penfield was stimulating that area of the brain, and you get the same thing. A person hears a voice that's not there. Has all sorts of implications. I used to study philosophy, and we can get into other discussions about what is consciousness and all of this, but that's not today's talk. And today we can even, and work by Mike Fox and the group, even use this resting state connectivity to look at whole networks and the ways in which patients with strokes can inform how these networks function, et cetera. Paranoid delusions in schizophrenia, it's another form of threat and fear, but in this case, it's social threat. And again, these systems are activated, these fear systems, anterior insula and amygdala and ventral hippocampus. But, and here you see this ventromedial prefrontal cortex that should be regulating things that's asleep at the wheel. And that pattern, it turns out we're seeing in a number of different psychiatric disorders where the limbic emotional is hyperactivated and reactive, and the higher order control regions are hypoactive and not doing their job to modulate. And that's a theme that we're really trying to unpack and understand within and across disorders. And again, psychosis and temporal lobe epilepsy with paranoia converges with similar sorts of findings. I guess another example, very briefly, depression is not just accentuation of the negative. People like Helen Mayberg and Wayne Drevitz have done seminal work in understanding the role of the amygdala and negative emotion in depression, and also the role of the subgenual cingulate in regulating all of this. We, and then subsequently others, were interested in what's called anhedonia. What's anhedonia? Again, if I could call upon Dr. Pine.
The lack of being able to feel pleasure in activities you normally enjoy.
Yeah, so it's the lack of interest or pleasure in usual activities. And patients very often with depression have that. The things that they used to like to do, go to the movies with their friends or whatever, they have no interest in doing, et cetera. And it doesn't give them any pleasure. Long story short, we did an experiment where we wanted to test the hypothesis that the brain reward region and motivation region was hypoactive and not functioning properly in patients with major depression, and even more so in those who had anhedonia. And that's what we're seeing here. We can even do fancy things like graph theoretical network analysis and all these multivariate analyses and show that in this area, the ventral striatum, the nucleus accumbens, which is the key reward node for the brain, only in patients with major depression and only in the positive emotional condition, is literally offline in terms of its connection to other brain regions. So we have the beginning of a way to quantitate and visualize and start to characterize these disorders. We can look at whole networks and how they interact. And without getting into details here, when patients with depression say they see the world through blue-colored glasses, that everything has a sad tint, literally what we're seeing here, and again, I can go through this offline with anybody, is that in healthy people, our cognitive brain networks are segregated from our emotional brain networks to a certain extent. Whereas in major depression, they bleed together, and literally emotion overwhelms the cognition and thinking. Again, in a neurologic disorder like Parkinson's, when patients have anhedonia, lack of interest or pleasure for different reasons, guess what? In scanning there, it localizes to the same region. Finally, in a whole bunch of different disorders, if you have a lesion in your orbitofrontal cortex, posterior medial orbitofrontal cortex, you're going to have impulsive behavior. Some of you may have heard the story of Phineas Gage. His skull and the tamping rod are in the Countway Library right here. Very famous story of a railway worker in the 19th century in New England who had this rod go through his brain, his skull, and he lived. And he went from a upstanding, highly responsible foreman and citizen in the community to someone who couldn't keep a job, who was unreliable, who was telling bawdy, inappropriate, jokes and things, and who was engaging in problematic behavior. This was huge because this then, and his doctor, Dr. Harlow, reported it. He said gauge is no longer gauge. It got into this whole issue of what is a person? What is and it showed people dramatically how even things like personality and morality, quote unquote, could be instantiated in the brain such that when certain areas were damaged, you had a marked change in those functions. And long story short, we studied borderline personality disorder, bipolar manic depressive disorder, and mania. This is a patient with an arteriovenous malformation in the orbitofrontal cortex. And in each case, when that region was impacted in that way and hypofunctioning and couldn't respond to projections of future consequences of your action and generate your action according to social mores, the person got into trouble because of impulsivity. So let's, and frankly, these days, and this is work by Andy Horn in the group at the Brigham, you could even use imaging to identify the exact pathways in these regions and networks and use brain stimulation and even predict based upon the exact place that you stimulate, whether the person, in this case with refractory obsessive compulsive disorder, where a certain network is implicated, is going to get better or not. And we can use brain modulation techniques these days. And Sean Siddiqui, also in our department, is doing that in depression, where he uses resting state brain connectomics and is able to tell, even in individual patients with specific subtypes of depression, where to do transcranial magnetic stimulation, which is a kind of neuromodulation that's non-invasive, unlike the deep brain stimulation with electrodes. And these days, we're even developing things like focused ultrasound in the brain to be able to modulate these circuits. You can also modulate them pharmacologically, and you can use imaging to identify patterns that may even predict antidepressant response. As some of you may know, if you give an antidepressant, it may take 4 to 8 weeks for a person to get better. They could kill themselves in the meantime. And then you're trial and error. Well, if we can develop brain pattern profiles that may be able to say this particular person, it needs this particular type of medication because of the chemistry and the regions involved, then we're starting to be like cancer, where you say this person has this type of cancer, and based upon the imaging and the genes and the hormones for breast cancer, in this woman, it's this particular mechanism that's gone awry. And therefore, we got to develop this particular chemotherapy or drug to target that particular signaling pathway and mechanism in her. And therefore, we can predict that she's going to have a 94% chance of getting better when you can match her biomarker with these tests, these mechanistic biological measures with her outcome even. Doing appropriate validation. And that's where we want to go in psychiatry as well. Again, not ignoring the psychological and focusing on the biological. And then finally, even cognitive behavioral therapy, psychological treatments, we can see affect the brain. And this is a patient who we studied before and after cognitive behavioral therapy with post-traumatic stress disorder. And you can see coronally, if you're used to looking at this, that when the parent, I'm sorry, this is schizophrenia with paranoid delusions. And when the paranoid delusion score went way down, you could see that they're engaging their executive dorsal prefrontal cortex medially and laterally. And in a corresponding way, there's a diminution of the reactivity of their anterior insula and amygdala hippocampal complex. So literally, the non-biological therapy is biological. It's working through the brain and vice versa. So what are we doing with all of this? We have all these fancy analytics and the convergent principle, okay? Perceptual, cognitive, emotional, behavioral, social, and executive functions and interactions can be localized and characterized in the context of neural nodes, circuits, and connectivity, which can be impacted by a range of idiopathologies resulting in what we might call circuitopathies, which then give specific clinical presentations and responses to treatment, within and across the full spectrum of neurologic and psychiatric brain-mind disorders. And we can develop models of how to integrate all of this, which I'm not going to bore you with. In the future, we might imagine, with your generation and the science, developing a multimodal biomarker space in which any given subtype of an illness or patient where they live in a certain cluster based upon these biomarkers may suggest or be correlated with an underlying mechanism that we can then target more specifically. And that way we can get to the point where we can combine the systems level neuroscience with the psychology and the genetics and neurodevelopment and start to even get to the point where we can develop tests for these biomarkers diagnostically, characterize these disorders biologically, treat them in a much more targeted fashion, even in individual people, personalized medicine, and the holy grail, which is prevention. If we can identify profiles that suggest that a 14-year-old is going to become psychotic when they're 16 or 18 with schizophrenia, and we can cut that off at the pass and understand that process, then that's a whole new world, and that's really what we're aiming for. This is, for sure, team work, and this is members of our lab, among other collaborators, and I'm really happy to take any questions. I know, this has been a whirlwind, but hopefully compared to an hour ago, you at least have a sense of some of the excitement and methods and findings in the field with an eye towards what your generation now can bring to it. So thanks. Any questions, either in person or online? I have no way of checking online, but I'll rely upon you for that, I guess.
I'll read out questions.
Yeah, that's
right
fine.
now, we don't so far.
Okay. Don't be shy. Yes.
I'll have a go. So, first of all, brilliant lecture. Thank you. And certainly very much more enlightened than what I was an hour ago. You mentioned stress being literally toxic to the brain and potentially resulting in disorders later in life, and even transmission to the next generation through epigenetic modifications. Have those epigenetics been characterized? How well is that understood? I'm just curious.
It's still nascent right now, but and that's not my field. That's where some of you who are in, who use those methods can really advance this work. But my understanding is, so don't trust me on this, and it's going to be imperfect if not wrong, is that the methylation of and modification of certain regions on genes that code for polymorphisms, functional polymorphisms that are associated with the neurotransmitters and the neurogenesis and the setting of these thresholds of these systems have been shown to be abnormal and transmissible compared to, so if you have women who are pregnant who either have a certain history of trauma or undergo, some real terrible thing that happened during their pregnancy, compared to control women who didn't have that, in larger numbers. And then you look at them, you look at their children, you follow their kids and see who develops what and what their stress reactivity is and cortisol, et cetera, and some of the cortisol modifying genes. That's my imperfect understanding of sort of where the field is now.
Thank you.
Yeah. Other questions? Yes.
Thank you for this amazing talk. I want to ask about the convergence principle. You mentioned that epigenetic, genetic, and neuroimaging can combine, and we can use this and predict a certain neurodevelopment things about psychiatric disorders. Is there any modulations for before clinical symptoms? Can we detect what pathways or what pathways can cause this neuropsychiatric disorder. So is there
Yes.
prediction models for that?
Yes, people now are starting to do longitudinal controlled studies, and they have to be carefully designed, and they can have confounds, all the things that you might expect. But it is the case that people with certain signatures of reactivity of this system, for instance, this fear system, are maybe more likely to be the ones, two people can go through a trauma, right? One person has PTSD or, up to 30% or, 5 or 6%, and then a spectrum develops PTSD. Another person goes through the trauma and starts a charity and, is not, their life is not destroyed. Like, what's the difference psychologically and biologically? And we can now start to look at that. Similarly, in terms of some of the, this is not epigenetics, but the looking at the genetics and the functional polymorphisms, that the serotonin transporter polymorphism, for instance, the 5-HT transporter, differences in short and long allele combinations in that can predict who's going to have more likely to have a depression or traumatic response psychologically. And that even correlates with amygdala reactivity on imaging as well. So you can start to see the convergence. And then the hope, this is just obviously a schematic that I did with my son would have done much better graphically. This is my little whatever. But the, where in the future, and with you all doing your work and advancing the fields, together with clinicians, and I really want to make a plug for translational work, that, you'll have a patient who has schizophrenia or something, and they will have, and we know schizophrenia may be 17 different diseases, right? Or 24 or 78, or, so we need to carve nature at its joints. But then, you have their imaging, you have their genetics, you have their epigenetics, you have the metabolomics, their proteome, their immunome. And then each of those, you then have really big cloud computing and algorithms. And at least the hypothesis or the hope is that a certain combination of those will be predictive. Now, in order to say predictive as opposed to correlation, then we need an independent, we need to derive a model, and now we're using ML and AI for that, actually. Derive a model and then test it in an independent sample to see if it predicts and if it accounts for the variance in a way that suggests causality, et cetera. And so longitudinal studies combined with ML and AI, and these different measures. And today I've given you only a taste of the systems neuroscience, but the exciting basic science that many of you are involved in is going to play a key role in this as well. Yeah.
I had a detailed question about the schizophrenia experiment. So the experimental design fascinates me. So something like the words for the amygdala, the threat one, I can see how consistently you show the same thing to a different group of people. But then for something like schizophrenia, where you're waiting for a hallucination to happen, I was just curious how that works.
This was 4 years of my life in London, 2 years in London, but 4 years of my life. There were no methods, and this is where the methodological development comes in, and I'll make a plug to you as scientists that you got to really know your methods. There's some people who might say, oh, you get this sexy color picture, press the button on the software and get a result, and the difference, like anything, between people who really know their stuff and not is the degree to which they really understand the methods that are being used and so forth, and are careful in interpretation and so on. Long story short is that there were no methods. Hallucination is subjective. It's like trying to take a picture of a ghost, right? Subjective, random or pseudo-random, right? Transient, fleeting. So long story short, and then the imaging methods, which at that time were PET, don't have the temporal resolution to even capture that, let alone to transduce that. So we developed a method to have a person press a button every time they heard the voices and release it when they didn't. And we did a whole bunch of validation and so forth to be able to capture the brain state during the button press. But then we had to control for the button press too, et cetera. And then subsequently, there are other ways to do it in a data-driven way and so on and so forth. But that But the plug there is not only to focus on methodology, but if a method doesn't exist to study what you want to study, don't give up. And hopefully you're in a lab where your mentor will allow you to do this. And I think part of it was that I went over to England with funding from Cornell, and so I didn't have to be anybody's postdoc. I could just do my own work, and I didn't have to do their experiments and spend 2 years thinking about this. But regardless of that, and depending upon the mentorship, the lab, if you want to ask don't just ask the questions that well, that's fine, but I mean, if there's a question you want to ask and the method isn't good enough yet, part of the fun and excitement is to then really get into the biology and the physics and see if you can develop a new method that can detect that and or answer questions about that or interrogate that. So that's what we did.
So fascinating. And so we have a couple of questions in the chat now. Maybe I'll one or two things now. So Ekaterina says, thank you for a wonderful lecture. You have mentioned self-awareness. I wonder how come self-awareness is such an important stepping stone for therapy of mental illness? What is it about neural systems supporting self-awareness that can have such an impact?
It's a great question. It's all about, in my mind, increasingly too, it's all about metacognition. Otherwise we're automatons, and this gets into a whole other philosophical thing, and we can talk about AI agents and everything else, which we're not going to talk about now. But the point is, and for any of this, and you can just reflect or see it in kids as they grow neurodevelopmentally, it's that meta level of when you're able to think about your own thinking and process with some mental distance your own emotions, that you can begin to have at least a conscious awareness of and ability to modulate and process those in a different way and not be a slave to them. So that's why that's such a key function, and we're so interested in these neural systems, and why, perhaps unsurprisingly, the work that we did with Dan Weisholtz showing that it was those regions of the brain that were predictive of who was going to get better with cognitive behavioral therapy. That was sort of confirmatory in that regard.
Thank you.
There's a question here.
Yeah, thanks for a great talk. I have a question, probably thinking about the currently medicine angle. There's some, for example, let's talk about depression, and you're using SSRIs on some patients that certain percent of them is responsive, but they're only responsive after a long period of time they're taking the medicine. From your experience working with these functional circuit level, I think the other way is all these targets of serotonin receptors or whatever is kind of broadly expressed in the brain, in all the functional circuits. How should we understand why drug works in a certain patient population and why it works so slowly? And how do you think about some of the theories of the resetting of the circuits from some of the neurological?
Great questions. A few different things, because you asked a few different questions I want to unpack in there. One is the drugs work slowly because they need actually changes at the neurotransmitter and synapse level, presynaptically and postsynaptically, that are adaptive to take place, and that has a different time scale than just the receptor being engaged. So it really is a plasticity of the synapse and the circuit that we're aiming for, and that has its own chemistry and genetics and time scale. In terms of the selectivity then, even when these things seem to be broad, that's where you get into subtypes of receptors. There's serotonin 5-HT1A, there's 5-HT2 receptors, there's 5-HT4 receptors. Each of these on autoradiography or with PET scanning, you can show are concentrated in different regions of the brain. And it's the confluence of those things that then will suggest that the nucleus accumbens, for instance, has a certain profile, which is enriched with D3 receptors or something like this. And so that's the targeting. And what we want to do to develop better drugs is to get more and more specific, so we have more and more on-target effects where we're modulating the function in the circuit and region we want to, but with fewer side effects by tickling the other receptors that that we're not interested in. Was there another question in there?
Yeah, the last question is, some people talk about like when you take these medicines, you kind of like reset the network.
Oh, yeah.
Like
Well, so now, I think ECT is an example of like really resetting the network. It's literally sort of like rebooting the brain. We can have a whole discussion about that. And ECT, electroconvulsive therapy, gets a bad name out of ignorance, really. It's probably one of the most effective treatments we have for very, very severe sorts of situations. The slide that I didn't go into gets into why, these sorts of things, some of our thinking about why these sorts of things may be taking place. But basically what you're doing, I think, is playing with thresholds and amplitudes and temporal profiles of stereotypic and plastic responses. It's a very abstract answer, but I think, one day, and again, with the aid of computers and stuff and the neuroscience, we're going to be able to model this to the point where, or test models where we can begin to understand that in a different way. But I think, these are very complex systems. And you'll see the word thermostat here, and that gets into this issue of resetting. There's resetting like ECT, like rebooting, but then there's I'm very interested in like bipolar disorder, manic depressive illness. How can the same person, the same brain, be wildly manic on one day, and then 2 weeks later, or maybe with a mixed state, 2 hours later, but be morbidly suicidally depressed? Well, that's their own brain is their own control. That's amazing. We can study that, right? And some people are studying, well, what's the manic brain look like? What's the depressed brain look like? And we've done that, but I'm really interested at this point in what's the thermostat? How does it switch from one to the other? Why is the thermostat off? Where is the thermostat? What are these regulatory systems? And that's why it's this frontal limbic modulation that I'm very interested in. And how does that break down, and how we can reset or stabilize the thermostat?
Thank you so much. We have more questions in the chat, but I think we're well out of time. I will save the questions and share with you.
Yeah, I'm happy to answer them, actually, each of them. So,
Okay.
if you just let me know later, and then we can, or people can email me, and yeah.
That sounds great. Thank you. We will send out the recording of the talk and your email website. So thank you so much to everyone for attending. Thank you for a wonderful talk. And if you're joining for the lunch, please come to the front.